Hello, and welcome to this session on the scientific method. As always, please make sure that before you start listening to this video, you've got the PowerPoint lecture notes in front of you so that you can take notes as you make your way on through. It's important that you do that so that you can hold up your end of the conversation and develop your understanding of this material just a little bit further when we're in the classroom. So let's jump right into it and pick up on the scientific method. As we've said in previous sessions, we have the scientific attitude as being central to how it is that science is conducted. And we've mentioned previously that scientists have an attitude that we might describe as being cautious or even skeptical. Not all the way to cynical, but cautious or even skeptical. And this is true relative to non-scientists. We might say, for example, that before believing a claim, scientists often require findings be replicated. So we've mentioned previously what it is to have a replication and why replication is important, and we'll have some further conversation on that even during this session. But replication is a very interesting topic on a few different levels. First, we can say that there might be some advantages and disadvantages associated with requiring replication, and what I'd like to do now is get your intuitions on that. So in just a moment, we'll ask you to stop the video and see if you can jot down maybe 2 sentences that address this issue. What are some pros and cons associated with requiring replication? I'd like you to give me 1 sentence on an advantage associated with requiring replication, and also 1 sentence giving me a cost associated with requiring replication. So we'll give you a moment to do that while you stop the video. Okay, welcome back. I look forward to hearing what you have to say about the pros and cons of requiring replication, which is typically what scientists do before they're willing to believe a particular claim. Okay, let's go on and have a related question that we'll ask you to address in just a moment. And this is that scientists understand evolution to be true. In a previous session, we talked a lot about evolution and how it lines up well with the generality, the originality, and the precision that are the virtues that characterize science and so forth. So we're going to ask now about this. Scientists understand evolution to be true. Is evolution replicable? Okay, so I'd like you to muse about that. Maybe a related question is, in what ways might evolution be replicable? And we'll have you stop the video in just a moment, jot down a response to that, and then we'll take your answers in class. So go ahead and stop the video. Okay, welcome back. I'm looking forward to having that conversation about the situation with evolution and replicability, and hear your views on all of that. In our classroom, what we're going to do is have some time spent on an interactive activity where we're going to have you learn something about the scientific method by having you contrast things that are relatively scientific with things that are relatively non-scientific. It might not be obvious here, but this is something like a spreadsheet. There are some white lines that I can see, but maybe are not coming through on the video. And we're going to ask you to cut and paste some items over here on the left side, which are shown in blue. There are some words and some phrases that we're going to ask you to cut and paste into the appropriate column and the appropriate row. So as an example, we'll ask you to see if you can take the word ambiguous, cut it and paste it into either this column on the far right for that which is scientific, this column which is next to far right, which is non-scientific, and we'll see which of the rows would best describe that particular term. And we'll mix and match these, and then we'll share our answers and see if we can develop some intuitions about how it is that scientific reasoning and the scientific method differs from our non-scientific, more everyday kinds of approaches to understanding the world. Should be a fun exercise. I look forward to doing it with you in class. Okay, so why don't we go on now and talk a little bit about our intuitions and remind ourselves that the scientific method allows us to get beyond what we know from intuition, what we know with respect to what just seems to feel right, to use a very informal phrase. Previously, we mentioned that the theory of evolution is very non-intuitive in many ways because it reminds us that we're in some way connected to an oak tree or to a bacterium. And that doesn't always sit well with our intuitions, with what seems obvious to us. And yet by using the scientific method, we actually come to that conclusion very compellingly. So in some ways, the scientific method is like a telescope or a microscope. Those extend our senses. The scientific method can allow us to get beyond what we seem to know from intuition and allows us to get to a greater knowledge base that we wouldn't otherwise be able to attain. Okay, sometimes our intuitions are influenced by cognitive biases of different varieties. One is a confirmation bias. And we might define a confirmation bias as the inclination to remember only supportive evidence being confirming evidence. Okay? We have a tendency to disregard disconfirmatory evidence. It's a very human trait to disregard disconfirmatory evidence. So we need to be aware of our biases. A confirmation bias is one kind of a cognitive bias. There are other kinds of cognitive biases as well. One is called an illusory correlation. And this is wonderfully demonstrated in a terrific paper by Susskind. Here's a simple overview of that paper. It was entitled Children's Perception of Gender-Based Illusory Correlations. Okay? So you might remember from Intro to Psychology all about correlations. We'll do just a little bit of an overview now. I'll turn this to a blank screen and we'll draw a couple of correlational diagrams on the board just to help you recall what correlations are all about. So here we go. We'll put that away. We'll blank it out just for the moment. So when we're talking about correlations, what we typically have is a plot that might look like this. We might call it a scatter plot. Okay? And we'll have some variable on the x-axis, a different variable on the y-axis. It might be something like one's level of education, which I will abbreviate by EDUC, and maybe the salary that somebody makes. And we can ask about a relationship between these two variables. I'll draw it three different ways. Okay, we'll use the same labels for each of these. You might imagine that we have a positive relationship, a positive correlation. You might imagine that we have a negative correlation, or you might imagine that we have, in fact, no effect. And if we were to put in some data points here, consistent with this best fitting line, those data points might form something of an oval shape. In this case, a positively sloped oval shape as we go from left to right. Over here, we might have the case of a negative correlation. Okay? And again, the data would tend to form a cloud that isn't circular, but something of an oval. In this case, the oval has a downward slope or a negative slope. And here, if we plot the same variables, we could have, in principle, come up with a data set that has no particular pattern, at least no linear pattern. And if we put enough dots in here, we might find something akin to roughly a circular pattern. Okay? So no correlation, a negative correlation, and a positive correlation. You might remember that from Intro to Psychology. Psychologists and other scientists as well are very interested in all different kinds of correlations. So let's take this away and see if we can get back to Susskind's illusory correlations in our PowerPoint presentation. Okay, so this title again was called Children's Perception of Gender-Based Illusory Correlations, and this came out in 2003. So this was a very interesting study. It was a correlational study, and what we were trying to do is understand to what extent we can find an association between two variables, just like what we had drawn a moment ago. But these two variables were gender on the one hand and action on the other hand. Okay? You might think of that action variable as actually being an activity variable. The two levels of gender were actually male and female, as you might imagine, and the two activities were interesting. They were reading and knitting. So here's what goes on. The researchers wanted to understand something of an illusory correlation. Could they demonstrate that even young children were inclined to see a correlation when in fact no correlation existed? And they used this 2x2 design. What they did was they showed pictures to children, and each picture contained one of the 4 2x2 possibilities in this design. So a picture might contain, for example, a man who is knitting or a man who is reading. It might contain a woman who is knitting or a woman who is reading. And they would show these in random order to each child. They would show them many, many pictures. Critically, they had exactly the same number of pictures in each of these 4 categories. After all these had been shown, they asked the children to report which kinds of category combinations had occurred most frequently. And very interestingly, it was the case that contrary to what you might expect if the children were remembering things properly, the children exhibited an illusory correlation. They erroneously believed that they had seen more pictures of women knitting than of men knitting, even though it was the case that exactly the same number of those pictures had been presented. Now, why is that the case? Well, presumably the children were operating with some kind of schema, right? This idea that they might have that takes the form of a stereotype, a widely held belief. And their widely held belief might have been something along the lines that women are more likely to knit than are men. Well, that might actually be true. Some stereotypes are factually correct. Some stereotypes are factually incorrect. It might be the case that the children had that stereotype and that was actually giving them a bias in their cognition such that they had an illusory correlation. They thought that there was some association between gender and activity when in fact in the stimulus itself it was not there. So they were seeing some kind of a slope, so to speak, when in fact the slope was physically zero. Okay? An example of an illusory correlation. And this is something that we would need to be aware of when we're engaging in any kind of scientific investigation. We need to be fully aware of our different biases. Something else we need to be aware of, and that is how it is that we interpret what we think we're seeing. And that is demonstrated here in a very classical sense by what you might remember from Intro to Psychology is the story of Clever Hans. Clever Hans is the horse, as you might imagine, and here we have this question of whether Clever Hans is capable of counting or not. I'd rather not say too much more about Clever Hans because I want to give students the opportunity to talk about this and develop this idea in class. You might choose to go on to Wiki or to look at some other source to figure out what was going on in the Clever Hans scenario and how does this relate to what we're talking about here in research methods. So I won't say anything more other than the question here was, can Clever Hans count? Okay. A related issue that we might have when we're investigating something like animal cognition. Can Clever Hans count? Can dogs do things like detect cancer? There had been some anecdotal reports that dogs were acting very differently toward their masters just before their masters had been diagnosed with cancerous tumors. So the thinking might have been that somehow maybe the dogs can actually smell a tumor or some of the related biomaterials associated with a tumor. Okay, so this led to some very interesting investigations, one by Willis et al. In 2004 and one by Gordon et al. In 2008. And there are some conflicting results between these two papers, and this is one of the ideas that we want to develop here early in our course, that sometimes science produces conflicting results. We don't always get a perfect replication. And then we, as scientists, have to make some sense out of sometimes differing kinds of outcomes. We won't go into some of these specifics right now, but what I have here is this PMID. That's the PubMed identification number. PubMed is a database made available by the US government, and it contains all kinds of medical and behavioral studies. And in this case, the issue was whether dogs can detect cancer. And during our class, we'll put in the PubMed ID. We'll look at the abstract from this paper and from that paper, and we'll see what some of the challenges might have been in this question, can dogs detect cancer? And how might that be related to some of the challenges that we see even back here when we're talking about Clever Hans. Okay, so this is a study that was going back to the early part of the 20th century. Here we are in the 21st century, and some of the challenges that we see methodologically, even from 100 years ago, will always be rearing their heads, and certainly that's the case here, and the question of whether dogs can detect cancer or not. Okay, let's move on, and we'll ask you in a moment to stop the video and do one other exercise for us. Here we have a picture. I'll try not to say anything more about that, but what I'd like you to do is stop the video and in one sentence describe what you see here. So go ahead and stop the video and do that, and when you're set you can turn the video back on. Okay, welcome back. I look forward to hearing what you had to say. What I can tell you is that I do this exercise frequently in the classroom, and students make lots of different kinds of observations. In fact, probably no two sentences that my students generate will be identical to each other. So here's one stimulus, and we get lots of different statements about what's going on in this picture. Describe what you see. Okay? Now, sometimes what happens is a student will say something like, oh, we have two friends here that are racing each other. Okay? Now that's not necessarily an incorrect statement about what's going on here, but what we would say is relative to what the scientific method typically allows, that statement that we have two friends racing probably goes a little bit too broadly. That is to say, scientists are very careful in their inferences. They avoid reading too much into the data. We do see what appear to be two men here. We don't know whether they're friends or not, and we actually don't know whether they're racing. Okay? Now, it could be the case that they are friends. It could be the case that they are racing, but those are inferences that are not directly supported by the information that we have here. If we do say that they are friends and they are racing each other, we've gone a little bit beyond the data. Okay? We would need more information before we could actually draw that conclusion. So this is a habit that we try to acquire, particularly when we're engaging in observational studies, also when we're engaging in experiments. When you see the data set, we need to be very careful to stick close to the data and not read too much into the data set. If we do think that these two men are friends and that they're racing, when in fact they are not friends and they are not racing, we've made what we might call a type 1 error. And what we mean by a type 1 error is that we have a false positive. That is to say that we've read too much into the data and we believe something to be true about the data that isn't warranted in the data. Okay? All right, let's go a little bit further, okay, and talk about something that we've mentioned in a prior video, but we'll just remind ourselves of it because it's so critical. Science relies on operational definitions, a description of a concept solely in terms of the operations used to measure it. So this might seem redundant, but I actually like the redundancy in this case. Operational definitions, we're stating the operations that we're using for measurement. Okay? Okay, let's do an exercise. Let's generate some everyday definitions of intelligence, and then let's also generate some operational definitions of intelligence, and we'll see how those two differ. So in a moment, I'll ask you to stop the video and create two sentences. In one sentence, give us an everyday definition of intelligence, and then we'll see if we can get to a more scientific operational definition of intelligence. Stop the video, and we'll be back in just a moment. Okay, welcome back. Let's go on to one other exercise. In a moment, we'll ask you to stop the video one more time and see if you can generate some responses to this. Generate an example of a measure that is valid but not reliable. And then, separately, generate an example of a measure that is reliable but not valid. So here we're trying to get very clear on the distinction between what is valid and what is reliable. You can stop the video, and I look forward to hearing your responses in class. Okay, welcome back. I look forward to hearing how you've distinguished validity and reliability. This will be a recurring theme in our course. Okay, another idea from our course will be, and you'll see this many, many times, we'll be talking about different kinds of instrumentation and what the instrumentation can do for us. Whenever we have an instrument, and shown here we have an MRI device, we can ask about the validity and the reliability of that device. To what extent are MRI studies valid and reliable? Okay, that's going to be an important question for us. Here we're using MRI as one particular device, but we can talk about other kinds of scientific instruments as well. We'll see some examples in a moment. One particularly relevant example of a scientific instrument for this course will be the survey. We don't always think of a survey as a scientific instrument, but it is a scientific instrument, and we can talk about the validity and the reliability of surveys, just like we can talk about the validity and reliability of, say, MRI measurements. Here's a potential pop quiz question. In your own words, explain how accuracy is different from precision. Also, generate your own example of an instrument that is precise but not accurate, and vice versa. So there's a lot going on here. We're actually asking you to do three things, right? We want you to first explain how accuracy is different from precision, and then we want you to generate your example of something that's accurate but not precise, and finally something that's precise but not accurate. This will be an excellent exercise, particularly since you've got to do all the generation. Okay? So we're asking you to generate responses here, and you can stop the video now. Okay, welcome back. I look forward to hearing your responses on this. We're talking about different kinds of instruments here in this particular shot, we've got the MRI device. There are other kinds of highly technical instruments. One I alluded to in a few videos ago was this device called the transcranial magnetic stimulator. And you see it shown here. We can ask, when this is used, about its validity and its reliability. We can also ask about its accuracy and about its precision. A very different kind of device where we actually take a magnetic pulse, which is measured in Tesla. That's going to be shown here on this axis. And we can see how the magnetic intensity is changing over time. Very interestingly, this is a 1 millisecond interval from this portion to this portion of the graph. And you can see that this is actually rising and falling over something on the order of microseconds, very precise in time, very small units of time. Okay? So here's transcranial magnetic stimulation, where we momentarily alter the state of the brain and we see how behaviors or thoughts or feelings might change in the presence of this disruption. Okay? Very interesting kind of device, very different from a survey, for example. But whether we're talking about surveys or MRIs or transcranial magnetic stimulation, we can ask about validity and reliability, accuracy and precision. And we'll continue to make those distinctions and get practice with that throughout our semester. Okay, let's move on to another important aspect of the scientific method, and that's the hypothesis. And we can simply define a hypothesis as a tentative explanation for something. It might be a tentative explanation for some phenomena that we observe in psychology. Earlier we used an example of depression. It might be obsessive compulsive disorder, whatever the case might be, a tentative explanation for something. Okay? And here's an important potential pop quiz question. Identify 3 conditions that make it impossible to test hypotheses. Okay? I look forward to your answers on that and having some discussion about that in class. You can stop the video now and give that a shot. Okay, welcome back. I look forward to hearing what you've got to say about the conditions that make it impossible to test hypotheses. Okay? Something that I have for you on this slide, I will not read at this moment. There's quite a bit of text here, and this is something that will be very clearly legible to you when you're looking at it on your computer screen, or even when you're looking at it on your printout. This is something from the National Academy of Sciences one more time. We had earlier looked at a definition of science from the National Academy of Sciences. Here, what we're doing is we're looking at how that academy defines fact, how it defines hypothesis, how it defines a scientific law, and how it defines a theory. And I won't go through all of these right now, but I'd like you to read them so you have some idea about how the National Academy of Sciences, anyway, a highly respected scientific body, uses these terms in a relatively precise manner. Okay, let's see if we can go on to talk about the typical 4 goals of the scientific method. I sometimes jokingly call these the Fab Four, and I would like you to memorize these. We're going to get lots of practice in working with these throughout the semester. The first and most basic of these is description. The next is prediction, and just a word about that here. Prediction is going to be used interchangeably with correlation. More on that later. Next we have explanation, and we're going to use explanation interchangeably with causation. Explanation and causation will go together. Finally, our ultimate goal is application. We're hoping that we can apply what we learn in science to solve some real-world problems. So these are the Fab Four, and we're going to discuss each of these in turn, starting with the one that typically we need to start with when we're investigating any particular phenomena in psychology, but this is largely true in the other sciences as well. We'll be talking about description. So here we go. We might say that description is described by the procedures researchers use to define and classify events and their relationships. Okay, that is what description is all about. Here's an example, the DSM. I'll pause just for a moment and ask you to think about this abbreviation which you've seen before. What does DSM stand for? Can you recall? Okay, here's a little hint. Okay, this is the Diagnostic and Statistical Manual of Mental Disorders, DSM, and then we get different numbers. This person happens to be holding a copy of DSM-4. Okay, we're up to versions beyond that at this point, and this is published by the American Psychiatric Association. And what goes on here is that the DSM will serve as a description for many different kinds of mental disorders. So for example, it might be the case that we're interested in depression, or we're interested in obsessive compulsive disorder, or OCD, and this constitutes a description of those disorders. To many people, the description answers a question of what. It answers a what question, whereas explanation answers the why associated with that phenomena or associated with that question. So here we're just describing what OCD is or what depression is. There's no attempt at this level of description to necessarily identify what the covariates are or what the correlations are. There's no attempt to say what's causing it, and there's no attempt here to say how to remediate the situation or how to make somebody's life better. That would be an application level endeavor. Here we're just describing what OCD is or what depression might be. Okay, that's purely descriptive work. On the topic of description, here are some dichotomies that might help us to more deeply understand what goes on when we are in the descriptive phase within the scientific method. Okay? First, we have quantitative descriptions and we have qualitative descriptions, and we use them all the time. Later on in this course, you'll see how we talk about these, and we're going to be spending quite a bit of time on quantitative analyses. Can you think of a variable that could be described either quantitatively or qualitatively? Why don't you stop the video and see if you can come up with an answer to that question. Okay, welcome back. I look forward to hearing your responses to that. We'll also now go on to make a distinction between another dichotomy when we're talking about description, and that dichotomy is the nomothetic versus the ideographic approach. Okay? Broad general rules that describe groups would be the nomothetic approach, and then when we're talking about individuals, we're using an ideographic approach. Maybe this will be helpful. You might have heard of the word idiosyncrasies. We might say that, oh, my friend has his or her idiosyncrasies. And when we say that, idiosyncrasy, we're referring to something that's very specific to that individual. We can take an ideographic approach to our description, and we can talk at great length about a particular person, for example, who has depression, or a particular person who has obsessive-compulsive disorder. Alternatively, we can describe large populations of people. And when we're talking about large populations, we might describe that data set in different ways. We might use some quantities, which would include things like means and standard deviations and so forth. That would be a nomothetic approach. Here is an example of a nomothetic description. It is a very interesting piece of work that was done a few years back where somebody was interested in describing the different paces of life in different environments and in different countries. So this person maybe had done some traveling and had noticed just very informally that in some nations we have a relatively fast pace of life, and in other nations we have a slower pace of life. And pace of life might be thought of as a variable that would be of interest to psychologists because it speaks to issues relating to behaviors, thoughts, and feelings, maybe particularly to behaviors. So we have to operationally define what we mean when we say the pace of life. These particular researchers, I hope it's visible to you, came up with 3 ways of operationally defining pace of life. They can measure walking speed, and they can measure that in seconds. And they can measure postal speed. How fast do postal clerks process their mail? And we can measure that in seconds. There are ways of defining that very precisely. Here's another interesting idea. We can measure the accuracy of a clock. Okay? You might imagine that in a society that has a very fast pace, people might be very aware of what time it is. In a different society, a different country, where there's a much slower pace, maybe people are less interested in exactly what time it is, and accuracy might matter less. So we can actually make measurements of clock accuracy and postal speed and walking speed, and we can begin to rank order different countries on the extent to which they have relatively low scores here, which are going to refer to faster times, and those who have maybe higher scores are going to refer to slower times. And we can rank them and so forth. It turns out that Switzerland had the fastest pace of life of these 31 different countries, and Mexico had the slowest, at least according to these 3 measures. Just to set the stage for something that we're going to learn a little bit later on, the way that these are ranked is by something called a z-score. A z-score is nothing more than a standard deviation. And this idea that Switzerland has a rank of number 1 and a z-score of minus 3.43, it would mean that Switzerland has a score that's 3.43 standard deviations lower than the mean on these 3 variables. If we go all the way down to Mexico, we get positive 4.23. That would mean that Mexico had very high scores, and again, higher scores on seconds would indicate that they had a slower pace. They were 4.23 standard deviations slower than the mean. Okay? So very interesting way of thinking about something like the pace of life, and very descriptive. Here this was a quantitative description. It's also nomothetic because we're describing not a particular individual, but we're describing larger groups of people. In this case, the unit of analysis is the country. Okay, let's see if we can now move away from our most rudimentary goal of description onto our second goal, which is prediction. Good descriptions can provide the basis for prediction, the ability to use one variable to estimate another variable. And earlier I had drawn some scatter plots, and we were reminded of these best fitting lines that are positively sloped or negatively sloped or have no slope whatsoever. Okay? So we're going to be using prediction and correlation interchangeably. Later on in the course we'll be talking about correlation and regression. Regression will be used for predicting. When particular scores on one variable tend to be associated with particular scores on another variable, the variables are said to be correlated or covary with each other. Okay? I'll give you a moment to stop the video and jot down a couple of correlations that we've not talked about in this video. We'll see if you can generate some novel examples of correlated variables. Okay, welcome back. I look forward to hearing your examples. Okay. Here's something that we hope you took away from Intro to Psychology. Correlation does not imply causation. That's such an important idea that we're going to make it a mantra for today. So I'll ask you to say it with me. Here we go, everybody. Correlation does not imply causation. There you go. Okay? That's an important mantra that we hope you take with you when you finish this course anywhere in life. It's tempting to think that correlation implies causation, but it really doesn't. Okay? Beware, some rigorous and sometimes even snooty scientists will sarcastically comment that a study has produced what they call compelling correlational evidence. This means that they don't really like the study. And what's going on in this scenario is that the scientists may have read a paper that was attempting to make a causal claim, and yet the researchers might have offered only correlational evidence. So they might say something like, well, the researchers have offered compelling correlational evidence, and what they're really implying is there's no causation going on in this study. Okay? Okay, so that's one of the ideas that we want you to remember. A couple of other subtle points. Humble scientists, those who exhibit some humility in the work that they do, will present causal relationships, and we'll talk about causation in a moment, with a dash of humility. Those variables are correlated, is what they'll say about their study, when in fact they probably could have made a causal claim. But remember that we said scientists are very cautious. They try not to read too much into their data. They might basically make an argument laying out all the evidence for a causal relationship, and then at the very last moment they say, we have a strong correlation here. Okay? And they do that with a wink and a nod. They're really hoping that you think it's causal, and they probably have enough evidence to do so, but they're merely stating that it's correlational. It's a more modest claim. One last complication that I think I'll share with you because I think you can handle it. Although it is true that correlation does not imply causation, we have this other fact, which is that some correlations are causal. It's just that not all of them are. So it gets to be a little bit tricky, but we'll get practice with this as we move through the semester. Okay. Beyond description and prediction, which we said is correlation, the third goal is explanation. And this occurs when causes are known. So we're using explanation and causation interchangeably here. And this is a very difficult thing to do. It's very difficult to make a causal claim. There are some philosophers who have argued you really can never truly make a perfectly causal claim. But in science we give it a try, and we get better claims about causation using particular kinds of procedures that we'll be talking about. Okay? So scientists typically set 3 criteria for drawing causal inferences, claiming that one variable causes another variable. Okay? And we're going to take these one at a time. Okay? The first criterion of causation is covariation. Okay? Covariation is necessary but not sufficient for causation, and we'll ask somebody to try to explain that difference to us. This is a subtle point, and some people are very comfortable in understanding the difference between that which is necessary and that which is sufficient. Okay? What we can say is that covariation is necessary. We have to go at least that far, but not sufficient for us to draw a causal inference or to say that we have causation. So we'll give you a moment in class to see if you can explain that distinction to us, maybe more fully than I've done for you here. Okay? Okay, the second criterion, after we've established that there's a correlation, is the following. The second criterion for causation is a time order relationship. Causes must precede effects. Okay, and I want to say a little bit more about that. This idea of time order relationship. It would be fairly easy for us to find two variables that are correlated very strongly one with the next. And maybe when we see that correlation, we want to draw a causal inference. But we actually can't draw a causal inference unless we understand the time order relationship. And here's, I think, a very obvious example of that. In modern society, there are a lot of debates about gun usage. And there's a lot of debate about gun usage and the rate of prevalence in different cities throughout any given country, certainly throughout the United States. Let us hypothetically say that there is a positive association between the level of gun ownership in a city and the level of crime in a city. Those two variables are positively correlated. If we were to plot gun ownership and the level of crime, there would be a positive relationship between those two. I'm not saying that's actually true, by the way. I'm saying we're going to take that as a hypothetical positive correlation. Okay? Well, we can have at least two different interpretations of that. We can say that those two are positively correlated to one another, and that what happens is that because there's a high crime rate, some people go out and buy guns to protect themselves. So in that scenario, the crime is causing the high gun ownership rate. But you could play the game in the opposite direction. That is to say that once there's a lot of guns in a given city, then the rate of crime, particularly violent crime, is going to go up because of the guns. So now we have a very different causal inference. Okay? And actually, in real life, in that particular scenario, we don't really know what's causing what, at least in most cases. Okay? So if we want to draw a causal relationship, maybe in an experiment, we have to be very careful about laying out the sequence of things and make sure that we can control the time order relationship. So far, we have two criteria down. There has to be a correlation, and there has to be a specifiable time order relationship such that the causes are preceding the effects. Okay? Okay. The third criterion for causation is the elimination of plausible alternative causes. This one might be the most difficult to establish. This is usually where there's the most debate and back and forth between an author of an article and the peer reviewers. Okay? The elimination of plausible alternative explanations. A reviewer of an article can always offer a cheap shot. They can always level a criticism by saying something like, couldn't there be some unknown alternative cause for the effect that you're showing in your experiment? And this is a cheap shot because the answer is always yes. There could always be some unknown cause. And this is why some philosophers of science would say we can never really get all the way to causation. Okay? Our burden as scientists, and the particular variety most relevant to this class would be psychologists. Our burden is not to eliminate all possible alternative causes. We're only required to eliminate those plausible alternative causes. That is our explanatory burden. And we typically can do that by having different kinds of control experiments, for example. Okay? So it's critical to remember that our criterion is merely to rule out and cross off of our list plausible alternative explanations, not all possible ones. Okay. Okay, to rule out plausible alternative causes, experimenters must eliminate confounding variables. Confounds would be a word that a lot of students use inappropriately. They learn the word confound, they learn that a confound is bad, and it is, and anytime they see a problem in a paper or a problem in an experiment, they'll say, oh, there's a confound there. Well, not all problems are confounds. Confounds are very specific kinds of problems, and we define a confounding variable here. Confounding variables are variables that systematically covary with the independent variable. Okay? Random errors in an experiment do not constitute confounding variables. Okay? Confounding variables threaten an experiment's internal validity. But before I talk about internal validity, I'm going to not go too far. I'll come right back here to what the confounding variables are. And we'll see if we can develop a very simple example that might make this a little bit more concrete. Again, the definition is these are variables that systematically covary with the independent variable. So let's see if we can generate an example of that. I'll give you this one that I think is simple to follow. Sometimes psychologists are hired as sports psychologists. And you might imagine that a sports psychologist is hired by an organization like Spalding, which makes sporting equipment. And Spalding might sell, for example, baseballs. And the folks who own the Spalding Corporation might want to know whether differently colored baseballs differ in their hit ability. That is to say that maybe a fluorescent blue baseball is more easily hit than is a white baseball. Okay? And so they want to know something about the colors of baseballs and to what extent they're hit. So we might have, for example, 4 different levels of an independent variable. We can have white baseballs, and we can have red, green, and blue baseballs. And we can measure on our y-axis how hittable each of those balls might be. Okay. So what we might do as an experiment is we might start out going to our local gymnasium where we can have an indoor pitching setup, and we throw some baseballs towards some batters, and we throw all the white balls first. Maybe early in the morning at 8 o'clock we start and we have some hitting of white baseballs. 2 hours later we come back and we do the red baseballs. 2 hours later we come back and we do the green, and then the blue 2 hours after that. Okay? All right, now what we have is a confound. We have something systematically covariant with our independent variable. Our independent variable is the ball color. It has 4 levels, and this is the variable about which we would like to know. We want to know if these differ from each other in hitability. That's on our ordinate or our y-axis. But we also did this at different times of day. Relatively early in the morning, we were giving the white baseball, and then later on in the day, we were giving different colors. So it might be the case that we do observe some differences across these conditions on our y-axis variable, our dependent variable, but it might be because of time of day rather than of color. So we have something systematically covarying, and we then have a threat to the internal validity of our experiment. So let's see if we can define now what internal validity is. Internal validity is the extent to which a study assesses what is intended to be studied. Here we're hoping that we can assess the hitability of baseballs, but we have this confound. We have a time of day confound in this particular case. A study can have internal validity, but not external validity. The extent to which a study's findings generalize to new populations, to new settings, and or new conditions. We mentioned that Darwin's theory of evolution has terrific generalizability, because it works not only on the Galapagos Islands, but really anywhere we find life on Earth. Okay? So we can think about the external validity independently of the internal validity. Poor sampling and poor operational definitions often generate poor external validity. Just to remind you of something that we had done previously, we have this research cycle. We start out with some real-world phenomena, and we abstract to a research representation by engaging in operational definitions and by engaging in sampling. But if we do those poorly, no matter how well we do our methodology, and no matter how well we do our data analytic techniques, we're probably going to have some difficulty in extrapolating or generalizing back to the real world if we've not defined our operational definitions well, or clearly stated our operational definitions, and if we've not done our sampling appropriately. Okay, let's see if we can now move on to the very last of our 4 goals, and this is application. So the final goal of science research is application. When someone describes the distinction between applied research and basic research, I'm very curious to hear how you think about that. I'm going to ask you to stop the video now and jot down what you think is the major distinction between applied research and basic research. It's okay if you have more than one idea about that. Okay, welcome back. Finally, in just a moment, I'm going to ask you to stop the video one last time, and I'll be interested in your opinions on the relative merits of applied research versus basic research. Earlier, we asked you to say just how these two are different from each other. Now we want you to elaborate on the relative merits of each of those. But before we do that, I just want to let you know that we're already at the end of our presentation, and this is our acknowledgement slide. And what we'd like you to do is take some notes about what you thought was clearest in this presentation. That's an important step, and also what kinds of things you would like greater clarification on, and hopefully we'll get some student-to-student conversation about each of those things when we're in class. See you in class.